Good afternoon, class. How are you? So, welcome to our microbiology 2. Um, our topic today is Introduction to Virology, and our specific objectives are three for today. The first objective is to define the term virology and the genome. Our second objective is to describe the core structure of a virus. And the last objective is to explain the viral replication cycle. So make sure you mute your mic so that we are on. And so we begin with the definition of terms. So our first term here is virology, and I want one of you to give us the definition of the term virology. Good, Sabina. Virology is the study of viruses. Then we have another term that is called replication. And replication here reverses the formation of the new infectious virions in sufficient quantities during infection in target host cell. For example, if the virus is HIV, then the host cell will be the CD4 T cell. We have another term called genome, which reverses the complete genetic information of a virus. And we say the genome of a virus can either be DNA or RNA. There is another term that we shall come across. It is called transcription and refers to the process by which a cell or a virus makes RNA copy of a piece of DNA. Then we have another term called translation, which refers to the process by which a cell makes protein using a genetic information carried in its RNA. So from there, class, I want us we look at the second objective, which is the basic structure of a virus. So all viruses, they share a common basic structure, which consists of the viral genome. And I say the genome can either be RNA or DNA, but a virus cannot have the two. So if a virus has DNA, it cannot have RNA. And if it has RNA, it cannot have the DNA. Then from that viral genome, it is surrounded by a protein coat that is called the capsid. So the viral genome together with the capsid makes up what we are calling the nucleocapsid. Surrounding the nucleocapsid is another layer that is called the viral tegument, followed by a second layer called the envelope. Not all viruses have the envelope. We have some viruses which are called naked viruses like the polioviruses, and they do not have the envelope. So emerging from the envelope is the envelope protein, which sometimes we call it the chippy proteins. So I want us to look at the functions of the viral parts. Who can give us the function of the nucleic acid? Good. So the nucleic acid can either be DNA or RNA, as you've said, and the function is that it carries genetic instructions. Leggett, I want you to tell us the function of the capsid. Come up again, Leggett. Okay, the capsid is a protein that is surrounding the nucleic acid of a virus, and the function is to protect the genetic material. Good. Then we have another part of the virus that is called the protein spike. I say this one can also be called the glycoprotein. For example, in HIV virus, it has the glycoprotein 120, which attaches itself to the CD4 receptor in the T cell. Then we have another part that is called the envelope. Envelope is a layer that surrounds the nucleocapsid, and some viruses do not have this layer, and therefore they are called naked viruses. Example of naked viruses are the bolioviruses, adenoviruses, and rhinoviruses. So we move to our last objective, which is to describe the viral replication cycle. So class, we have the 6 steps of viral replication cycle, and I want somebody to give me the first step of replication in the viral cycle. Could one boy, the first step is attachment, and attachment is the first step during the viral replication cycle. Sabina, what is the next step after attachment? Good. The second step is penetration, which we also call the entry of the virus into the host cell. Then, Agnes, I want you to tell us the third step in the viral replication cycle. Good. And coating. Um, so the fourth step is replication. The fifth step is assembly, and the last step is release. So I want us to look at each of the steps. The first step, we have said it is attachment. So in attachment, this is the specific binding of a virus to a specific receptor site on the host cell membrane using the proteins that we say we're calling the glycoprotein or the protein spikes. So here, each virus has a specific protein spike that is attaching or using it to bind to the host cell. Then our second step is penetration, which reverses the entry of a virus into the host cell. And the entry of a virus into the host cell can occur through the following processes. One, it can occur by receptor-mediated endocytosis. For example, in case of HIV, it can occur through that process. Then you also have the fusion, the membrane fusion, whereby the membrane of the virus and the fuse with the membrane of the host cell. The third step is uncoating. Sabina, what do you understand by the term uncoating? Good. So this is where the viral capsid is removed, and this can be by degradation by viral or host enzyme or by symbol dissociation. This is essential for release of viral genome into the host cytoplasm. The fourth step is replication, which refers to the formation of the new viruses or the new virions in large quantities inside the host cell. So the viruses will replicate in different parts of the host cell depending on the kind of the virus. If the virus is RNA, then replication will take place in the cytoplasm. And if the virus is DNA, then replication will take place inside the nucleus of the host cell. Our fifth step is the assembly or maturation, whereby we shall have the packaging of the proteins which are newly replicated inside the host cell together with the genome so that we have the complete viral particles. And our last step here is the release, whereby the newly synthesized viruses are released from the host cell, either by causing the cell to break apart in what we are calling the lysis, or waiting for the cell to die, or it is through padding off through the cell membrane of the host cell. So those were our today's objectives, and I hope you have understood. Anyone with a question? Okay, so having no question, then I will leave you with an assignment. Our assignment for today is to describe or to classify various viruses. Describe classification of various viruses, and that marks our the end of our lesson today. See you in the next lesson. And if anyone has a question, we shall answer in the next lesson. Goodbye.